Okay. Then there are several ways like in which microorganisms are harmful for us as well. Can you quote some of them from our daily life?
Like coronavirus.
Yeah, good. So basically, they make us sick.
Yes,
Huh?
sir.
We see that uh, food gets spoiled, food poisoning. Hmm? Food poisoning is also because of several microorganisms, particularly
Yes, sir.
bacteria and fungus.
Fungus.
So it can lead to food poisoning also. So microorganism has both advantages while there are some disadvantages also. So that's what we would be studying in the chapter. Also, we would be learning first of all about different type of cells also. Okay, now because in the chapter many times we'd be getting to hear these terms like prokaryotic and eukaryotic. So thereby you need to understand them first. By the way, this
Okay.
chapter hasn't been started in your school as of now, right?
No, sir, it hasn't.
It hasn't started. Okay, then. Also, whenever your test marks are out, whenever you get your marks of the test, make sure that you share it with me.
Okay, sir.
All right. And also, I would like to advise you that even in the when your exams are going on, I mean to say that you since in class 8th standard, so thereby you need to uh, prepare for your exams early, so that even if the exams are near and let's say a different chapter is going on in the batch, so thereby you can
Yes, sir.
attend those classes also. Getting it now? What I'm trying to say.
Yes, sir.
Otherwise, you'd be missing on important things.
Okay, sir.
All right. So without any further delay, let's start the class. Has anyone else joined the class today?
No, sir.
It's only you. See, in the chapter, you'd be learning about what are microorganisms, how we classify microorganisms, and what are the characteristics of these microorganisms which we are going to study. What kind of disease do they cause? Hmm? What kind of disease do they cause? What is food poisoning? How alcohol is manufactured? How in baking industry we get to see that microorganisms are used. Huh? So these are the things which we are going to use. We would be learning about vaccine. We would be learning about antibodies. Okay, no? So these are some of the things which we would be learning in the class. So before we continue further, first of all, you have a look at this slide. In this slide, you can observe you have a microscope, the
Yes,
tool
sir.
that is used to view microorganisms. So microorganisms are so small that we can't see them with our bare eyes. So thereby we take help of this. With the help of microorganisms, with the help of microscope, microorganisms are magnified several times. It can be magnified up to this many times. See, it can be magnified up to lakh to 10 lakh times. So that is the level of magnification in this. So microorganisms are the organisms that can't be seen with your naked eyes. Further, they are usually unicellular in nature. It's saying usually. It doesn't mean that all of them are going to be unicellular. There are
Okay, sir.
some that are multicellular as well. So that is one of the common characteristics of all the microorganisms. There have been several scientists who had who had great contributions in the field of in this field of studying microorganisms. So in biology, you have a field called as microbiology, where we get to study about microorganisms. Okay, no? So microbiology is the field where we study about microorganisms. So there was a person named Anton von Leeuwenhoek. Anton von Leeuwenhoek. So he has been called as father of microbiology. The reason being that he discovered bacteria and another microorganism called as protista for the first time. Also, earlier what happened as of today we have got so many literary sources, we have got so many research papers. Say for example you want to study a topic, say for example you want to study about elephants. You want to study about elephants. So you would be having so many research papers, lots of studies done on elephants. What are the different species of elephants? Huh? What is the population of elephants? What is the population of elephants? What are the behaviors of elephant? Huh? How the elephants evolve throughout the time? What is the geographical division of element, in which part of the world they are found, etc. and etc. So you would be able to take help of lots of research papers, lots of books are available today. But earlier what happened? Earlier you go back to 200 or 300 years ago, at that time there were few scholars, few scientists, and not much work was done, neither in the field of biology, physics, or neither in chemistry nor in other fields. So much of work was not being done. So at that time, the scientists like this, like Anton von Leeuwenhoek, out of many, he was one of the scientists who is kind of a self-proclaimed microbiologist, meaning that he would be doing research, he would be making observations, and then he would be deriving conclusion from it. But as a student, now what you would be doing in order to study something, say for example, you want to study a leaf of a plant, say you want to study a people leaf. Say for example, you are a medical student and you want to study about this people leaf. So you would open the research papers. If there are some research papers done on people leaf, what are the characteristics, what are the type of veins found in it, what is the density of stomata found in it, etc., etc. So you have available research papers, but earlier scientists had no such things. They would be, he would, what this Anton von Leeuwenhoek would do, he would take out this people leaf, he himself would be studying about it, he would be making observation and then writing it in a book. So he was kind of a self-proclaimed microbiologist, meaning that he didn't had much research papers or other resources at that time, and still he managed to give so many contributions in the field of microbiology. That's why he has been called as father of microbiology. Are you getting at what I meant to say?
Yes, sir.
Furthermore, there are different types of cells also. See, mainly there are two types of cell on the based on the differentiation seen inside the cell, meaning that if you were to look at the first cell and the second cell, look at this eukaryote and the prokaryote. You tell me, inside the eukaryote and prokaryote, the structures of which cell is more organized? Is it prokaryote or eukaryote?
I think it's eukaryote.
It is eukaryotic now.
Yeah.
So if you were to look at, by the way, just like in our body, we have got an organ called brain. Similarly, cells also have a miniature version of brain called as nucleus. So
Oh, okay.
nucleus, you can spot nucleus in this eukaryote, this dense structure, which is almost circular, a well-defined structure. That is nucleus, this whole structure that you see over here. Let me mark this. This whole structure is the nucleus. And you
Okay,
would
sir.
observe that it is well defined, it is covered with a boundary. But then again, look at the nucleus of this prokaryote. Is this well defined, well structured?
No, sir.
No. Furthermore, look at the size also. Size of eukaryote will be much larger, although in this image it is not that much larger, but usually it is much larger than the prokaryotic cells. Okay, no?
Yes, sir.
So roughly the size of this will be around 10 micrometer to 100 micrometer. Micrometer is a very small unit of length. We have units like millimeter, centimeter, no? Millimeter,
Yes,
centimeter,
sir.
meter, kilometer, like that. So micrometer is very small unit of length. So that you would be using to measure microorganisms, because you can't use millimeter to measure the length of microorganisms, no? Or the cell. So you have to take a very small scale like this. Hope so far it is clear. So complex, on the basis of complexity, eukaryotic cells are more complex than the prokaryotic cells. The nucleus is well defined, while nucleus of prokaryote is not well defined. Okay, now hope you are getting this concept.
Yes, sir.
Also, we see that in eukaryote, now this nucleus is like it's a true nucleus, meaning that it's a well developed true nucleus. While in this now it's a false nucleus. It is not actually a nucleus, but kind of looks like a nucleus and have got some characteristics similar to that of a nucleus. Okay, so that is the type of cell based on their complexity and based on the type of nucleus found in them.
Okay, sir.
So prokaryotes were the are one of the most ancient groups of living organisms. Those organisms in which this kind of cell will be found, they will be called as prokaryotes. Call them as prokaryotes. So it will be found in bacteria. Particularly, this prokaryotic cell will be found in bacteria right now. So bacteria's their structure is not very well defined. Even if you were to say, for example, you were to look at fossils that dates back to almost billions of years ago. So when you will be looking at those fossils now, so in them, this kind of cell will be present. Hope you are able to understand.
Yes, sir.
Okay. Then do tell me, are human cells prokaryotic or eukaryotic? Which one is true out of these two? Hmm.
I think it's
Human cells
you can.
also, it's eukaryotic now. Also,
Yeah.
you can understand it in this simple manner that eukaryotes are the modern cells. Modern cells. These are the older, or basically it would be
Cells.
proper to say them as ancient cells. How much
Okay.
ancient you see? Like even if you were to know, the oldest known fossils of prokaryotic cells or bacteria are found. You know, this is not just a number. Like, I mean to say that this there are some rocks now that dates back to 3.5 billion years ago, and in those rocks, fossils are usually found stuck between the rocks, inside the rocks now. Their imprints are found on the rocks. So oldest known fossils of prokaryotic cells or prokaryotic bacteria are found in the rocks. So when it was studied, it kind of looked like this. I have got an animation of it also in this one. That is how a
Okay, sir.
prokaryotic cell looks like. So hope you have understood that concept. By the way, let me erase this thing. So right now we are going to learn about various groups of microorganisms. So there
Okay,
are
sir.
5 groups of microorganisms. They are bacteria, you have protozoa, then you have viruses, you have fungi and algae. How many of them are there? 5.
5.
They are as follows. Major groups of, in short, microorganisms can be called as microbes also. So if it's written as microbes, don't get confused in that. So the first is bacteria. Second one is viruses. By the way, in your school, which book you are following, Vaiga? Which book
So
is
I
being
think
followed?
it's an
It is
NCRT.
NCERT.
NCRT.
Okay, okay. Apart from NCERT, no other books are being followed.
I think so. I don't know, sir.
You don't know? Like, have you not been going to school regularly?
I mean, yeah.
Mhm. Okay, okay. Anyways, you yourself follow NCERT or any other books also?
No, sir. Only NCRT, like school textbooks.
School textbooks. Okay, okay. So these are the major groups of microorganisms. So microorganisms are classified into 5 groups, namely bacteria, virus, protozoa, fungi, and algae. Make sure that you memorize them. Okay. By next
Okay,
class,
sir.
you should have memorized it. Now, microorganisms, they may be unicellular or may be multicellular also. Okay, now
Okay,
they
sir.
may be unicellular or multicellular. So that is microbes. Now you tell me that bacteria, singular and plural. See, note this thing down that singular of bacteria is bacterium. Singular of bacteria is bacterium. So if you were to have just, let us say that you are doing some experiments in the laboratory and you have only one bacteria, so that would be called as bacterium. Bacteria is the plural. Okay, no? Bacteria is the
Okay, sir.
plural. Hope you are getting it. Also, it would be better if you make sure that you take on notes also. Like I told you about this person, Anton von Leeuwenhoek. You will not
Oh, yes,
find
sir.
it in NCERT. So that is very important thing. As a general knowledge also, that is an important thing. So make sure that you note down his name. Like Anton von Leeuwenhoek is known as
Okay,
the
sir.
father of microbiology. Now further, what about this virus? So virus is the plural. Like viruses is the plural now. Its singular is simply virus. Okay. Further talking about this, like protozoa. So protozoa is the plural. In singular, it is protozoan. For fungi, it is like talking about this fungi. The singular of fungi is fungus. Okay, na? And that of algae is alga. So all this which I have written over here, that is plural, and in the next side it is singular. So bacteria in singular, it
Okay,
is called
sir.
as bacterium, virus is virus, protozoa, protozoan, fungi, fungus, algae, and that is alga. Okay, so do tell me, how are you going to be making notes? Will you be uh, I usually recommend the students to take live notes during the class only.
Yes, I take live notes.
Okay, you noted it down?
Yes, sir.
Okay, okay. Unfortunately, today I have been like trying for the past 2 hours to turn my computer on, but due to some issue in it, I couldn't turn it on, and thereby I won't be able to turn my camera on. Anyways, have you done writing it down?
Yes, sir. It's done.
Done, good. So let us learn about the bacteria. So bacteria are basically, they are unicellular and they are prokaryotic. So they are just made up of one single cell. Now just imagine how small a single cell is. Just imagine it. Try to think about it. Like um, just like for example, we look at our body, uh, and just you look at our body, and then think of this bacteria. Compare the size of a bacteria and compare the size of our body. How small a bacteria is. It is just made up of one cell and it is able to carry out all its process, all its function. Just like we go through go through different stages of nutrition, we ingest the food, we digest the food, we absorb it, then assimilation takes place, and then digestion takes place. In class 7 you would have studied about that. Yeah. So the
Yes, sir.
bacteria is also able to carry out pretty much all of the functions right now. So this is unique in itself. It's a unique thing. So bacteria are unicellular and they are prokaryotic organisms. Furthermore, they are going to be useful, they would be harmful as well. But the important things about the bacteria, which I'm going to tell you, is as follows. First and foremost, is that they are found in large numbers. They are found in large numbers. It's not like they would be found alone, they would be found in individual. They would be found in the form of colony. So they live like colony. Okay, now they are found in a colony. Furthermore, where they would be found? So they would be found in air It would be found in water. It would be even found in soil. It would be found on surfaces of different things. It would be found on the body and inside the body also. So, for example,
Sir,
yes.
I'm actually not able to turn on the camera because I'm actually joining from my phone.
Okay, no issues then, no issues.
Okay, so thank you.
You see, they are found in air, water, soils. Then it will be found in surfaces of different things, surfaces of objects. Like for example, the table the chair you are sitting upon. On that also there will be some bacteria. On our hand also there are so many bacteria, although we aren't able to see them. Inside our body are also there are some bacteria. Some of them are harming us. Of them are important for us. Hmm? Do you know this, that how many bacteria are there in the human body?
No, sir.
Guess a rough idea. A rough idea. Like,
Like
will it be? Yeah.
more than
Mhm.
1,000, like 5,000.
No, no. It's in trillions.
What?
It's in trillions. Note this important fact down, that there are about 38 trillion. How many? 38 trillion bacteria inside the human body. Getting it, Vaika? You might
Yes, sir.
find it very surprising and a very surprising fact, but that is true. Okay, now 38 trillion. While if you can recall, many times in the class, I would have told that the average number of cells in the human body is how much? 30 trillions.
Yes, sir.
There are 30 trillion cells in your body. So do tell me this important this thing, that the number of bacteria is more than the number of cells in your body. Getting this thing?
Yes, sir.
By 8 trillions, 8 more trillions of bacteria are in their body. But if you were to calculate the total mass of this many bacteria, that would be roughly around 0.2 kilogram. So since they are very tiny, they are very small right now. Although their numbers would be there's a big number, 38 trillion bacteria are there in your body, but their mass is only 0.2 kilogram right now. So there is a kind of close ratio of 1 is to 1. 30 trillion cells, 38 trillion bacteria in the body. So hope you have understood that thing. Furthermore,
Yes, sir.
you know about cell wall, usually found in the plant cells now. But the thing is that bacteria also have cell walls. So bacteria have cell walls too. Okay, now. So hope you have understood it. Also, you're writing it down also, no? Keep writing it down also. So we learned that bacteria are very small. They are single cell microorganisms, and they have cell walls. But they are prokaryotic because they do not have an organized nucleus. They do not have organized nucleus. Further, if you were to look at the size of a bacteria and that of a virus, so bacteria would be bigger than virus. Bacteria are larger than virus. They are larger than virus. That's why you'd get to see that viruses can enter inside the body of bacteria. Virus can make human body also a host, and they can make bacteria also a host. Okay, now viruses acts as parasites in that manner. Even the bacteria, many
Yes,
of the
sir.
bacteria are basically acting as parasites by making our body a host organism.
Yes, sir.
So say this is the size of bacteria, this is the size of virus. So viruses can go inside the body of bacteria since they are bigger. Then furthermore, bacteria would be of different shape and different sizes also. Some of the bacteria would be benefiting us, some of them would be harming us. So they are harmful and useful both. Okay, can you think of some common examples of bacteria? What are some common examples? Yes, Faika? Give some common examples of bacteria. You have so many.
Rhizobium
Yes.
bacteria
Rhizobium, very good.
and lactobacillus.
Lactobacillus. Then you have some more like salmonella. Huh?
Oh, I don't know, sir.
Okay, noted down also cyanobacteria. Cyanobacteria, or you have Vibrio. Then you have Mycobacterium also.
So spirilla.
Spirilla, very good. Hmm, Spirilla. There are so many. The list is so long. Okay, no?
Okay, so
Okay. Have you noted it down?
so 1 minute.
Okay, okay.
Yes, sir, done.
Good. So far what we have learned over here that bacteria are just made up of one cell. They are unicellular. They are prokaryotic because they do not have an organized nucleus in them. Then they are found in large numbers, meaning that they would be found in colonies. They can be found in air, water, soil, on surfaces of different things, on the body, inside the body. We learned about this interesting number 38 trillion bacteria are found in the body, roughly makes up 0.2 kilogram of weight. And another interesting feature is that they have got cell walls also. They have cell walls too. They are larger than the virus. They are harmful and useful both. Furthermore, on the basis of the shape, on the basis of shape, there are four major types of bacteria. On the basis of shape, how do they look like? You have four main types of bacteria, namely Bacillus, Vibrio, cocci, and Spirilla. Look at the next slide over here. Here you can observe the four types of bacteria based on their shape. The first bacteria, that is this one, looks have a shape resembling comma. Comma kind of looks like this. Let me just say like this now. So it kind of looks like comma. Getting it?
Yes, sir.
So that is a comma shaped bacteria. One such example of a bacteria that has this shape is Vibrio cholerae. And later on, while you would be learning about the harmful bacteria, you will get to know that this Vibrio cholerae will cause a disease called as cholera. Okay, no?
Oh, yeah, okay, sir.
Mhm. Then you have a bacteria having a spherical shape. How a saucer ball looks like, or how a cricket ball looks like. It has a spherical shape now, like this. So
Yes,
some
sir.
bacteria would be having a spherical shape, and they are found to be living in colonies. Colonies meaning that in groups. So that is called as cocci bacteria. One such example is Streptococcus. That is
Okay, sir.
Streptococcus. Then furthermore, there are some bacteria having a rod-shaped bacteria. They are called as bacilli. They are called as bacilli. One such example of a bacilli is bacilli or bacillus. It's all about singular and plural. Sometimes it is bacillus. So bacillus is plural, bacilli is singular. It's only the
Okay,
difference
sir.
of singular plural. Don't get confused with that. So one such example for bacteria having a rod shape. It kind of looks like a rod shape like this right now. So that is salmonella. One such example of that bacteria is salmonella. Another examples are like E. coli also. E. coli is also one such example. Then you gave an example of like a spirilla having a spiral shape, huh? Having
Yes,
a
sir.
spiral shape. So one such example of a bacteria having a spiral shape is Borrelia, and those bacteria that are having a spiral shape, they are called as spirilla. So these are the types of bacteria and their examples as well. So hope you have understood it. Note this down also.
Yes, sir.
And you have to memorize these things. So far, whatever we have discussed, make sure after every class you memorize them.
Okay, sir.
So we learned that bacteria are microscopic living organisms that have only one cell. And if you have just one bacteria, it will be called as bacterium right now. And bacteria can be found all over the world, including in your body. They are on your skin, they are in your mouth, they are in your digestive system. It is almost everywhere. And we have seen that the number of like if you were to talk about the bacterial cells, they are more than the number of cells in your body. Although Vaiga, most types of bacteria are not harmful. Some are even good for us.
Yes, sir.
Like for example, some of the bacteria are located on your skin. They are actually good. Some of them are found in your gut basically in your digestive system. So they are important there also. So gut bacteria keeps you healthy. There are some gut bacteria that keeps you healthy. You understand what does gut mean? Gut basically refers to the digestive tract, like stomach, small intestine, large intestine. Okay. So there are some gut bacteria which are keeping you healthy. How? Gut bacteria, they can break down, they can break down and absorb the food. Break down, help in breakdown of food, and also in some manner help you absorb the nutrients from the food. So that is the thing. But certain types of the bacteria would be making you sick also. So those types of bacteria that would make you sick, they will be called as pathogen. We'll be learning about that later on. Do let me know once you are done writing this. Yes, Faigar, done.
Yes, sir.
Okay. Let me just connect it to the charger. So see, as we know that some of them are going to be beneficial for us. Apart from medicines, can you think of some other uses of bacteria? Like you
Like
answered, yes,
like some food, like foods,
some for.
like the foods, yogurt.
Yeah, okay, okay. Food you are saying, yeah. Like
Yeah.
yogurt that contains the lactobacillus bacteria. Huh? So they are useful in a number of ways. Let us learn how we are using the bacteria.
Okay, sir.
So they are, right now we are talking about uses of bacteria. So you have in the pharmaceutical industry, it is used. Pharmaceutical industry basically here we are talking about manufacturing of medicines. So there it will be used. It will be used in making curd. How is curd made? Yes, how is curd made? Yes, Figo.
Uh, like keeping milk for a few days.
See, basically, actually, making of curd happens due to a process called as fermentation. See, basically, what is basically happening in this case now? See, you will take a small amount of curd. In order to make curd, you need curd, right now. In order to make curd, you need curd. So you'd have
Yes, sir.
seen your mom, like she would be taking a small amount of curd and she would be adding it in milk, and will leave the milk undisturbed for over a night. And next day, the whole milk turns into curd. So you take a small amount of curd. This will basically contain the bacteria. That bacteria, we all know that is lactobacillus. Now, so curd will be made from milk through a process called as fermentation. So first of all, let me explain this one also. But first of all, write this equation. Let's write this equation. So you have added some of it into milk. So it will be converting, like in the milk, lactose is found. Lactose is found in the milk. Upon adding bacteria, upon adding this curd to the milk, what happens? This lactose is converted into lactic acid. So lactose is converted into lactic acid. All right, now lactose gets converted into lactic acid. That's why curd
Okay,
is
sir.
having a sour taste. The sour taste in the curd is due to this lactic acid.
Okay, sir.
Then this lactic acid will coagulate the protein found in the milk. You know what is coagulation?
No, sir.
See, coagulation basically means that something which is liquid, it will become semi-liquid. Like when you look at milk and curd, milk is liquid, curd is kind of semi-liquid. We observe that thing, isn't it?
Yes, sir.
So that is called as coagulation. Or you would observe this, you would have observed this, like whenever we get a wound or cut, blood will slowly start to coagulate now. Initially, blood will come out from the wound. After few minutes, it will start to coagulate. Right now, that it starts to coagulate. So that is what coagulation is. Hope you're getting it. So this lactic acid, what does it do? This lactic acid basically coagulates the protein found in the milk and thereby results in the formation of curd. Also, you would have heard like curd is a good source of protein.
Yes, sir.
Okay, no? So let me write it over here. Lactic acid coagulates the protein in milk and thereby results in formation of curd. Hope you have now understood how basically curd is formed. Let me
Yes,
repeat
sir.
it again. It is formed through a process of fermentation where bacteria is used to convert the lactose found in the milk into lactic acid. And this lactic acid will coagulate the protein in milk and will result into formation of curd. So
Okay,
that is
sir.
the whole process. And this process is called as fermentation. This is a kind of fermentation reaction.
Yes, sir.
You have noted it down?
Yes, sir.
Good. So that is one such use of a bacteria. So we have simply used a starter, a small amount of it. Also, you know that you have to adjust the temperature. Like if the milk is cold, you have to warm it. You have to warm it slightly, right now. And you have like you'd see that. Have you ever tried making curd yourself?
No, sir.
No. So you can do this. You can try this doing this one. What you are going to do, you can take a bowl. You take a bowl, take some milk in it. The milk should be slightly warm. Then add this starter. Add some small amount of curd into it, and you can cover the bowl with a with a with a cloth. With a cloth, you can cover it and place it in a dry place and leave it overnight. You can even place it in the refrigerator. the next day it will be ready.
Okay, sir.
Okay. Furthermore, there are some other uses of bacteria also. Can you think of some other uses?
So rhizobium bacteria, it's used for like plants,
Hmm? To carry out what?
like to take nitrogen from the atmosphere.
Basically, it helps in nitrogen fixation. No?
Not nitrogen, from nitrogen.
Exactly. So it helps in nitrogen fixation. So the rhizobium bacteria which you're saying, this will be found in leguminous crops, no? Leguminous plants, like you have peas. So in the roots of the peas, we have learned that they have got bulb-like outgrowths called as, what do we call these outgrowths? Can you recall this? We have learned this previously many times.
Yes, sir.
That is called as root nodules. And in those root nodules, a bacteria is found to be living. That bacteria is called as rhizobium bacteria, as we told. What this rhizobium bacteria does? This rhizobium bacteria is capable of converting atmospheric nitrogen into useful compounds, into soluble compounds that the plant is able to absorb. Getting it? Let me write it on the next slide.
Yes, sir.
Rhizobium bacteria converts the, what is the percentage of nitrogen in the atmosphere? Yes, Vaiga?
So what?
What
Can
is
you
the,
repeat it again?
yeah, what is the composition of nitrogen in our atmosphere?
So I don't remember.
How can you forget that? It's such a simple number. It's 78%.
Oh yeah.
78% of nitrogen. Okay. Anyways, but that's a general fact. You should have remembered that already.
Okay, sir.
Anyways, okay. Furthermore, it would be used in making biogas also. Bacteria would be making in used in making biogas. Do you know about biogas? Biogas is uh, basically a gas that comes out after decomposition of biodegradable wastes. So
Yes,
it
sir.
can be used in making biogas also. Furthermore, it can be used in killing the plant pests. uh, It can be used there also. So there are different uses of it. Let us quickly summarize all of them. Okay, okay, na? Furthermore, there was this cheese also, no? Making of cheese. So
Yes,
in
sir.
making of cheese also, it will be used. Now let us understand that how bacteria are used in making cheese. Let us understand that thing. See, so there would be some certain kind of bacteria. For example, there would be like lactic acid bacteria would be used in making of cheese. So they would be again, uh, they would be using this bacteria in order to make cheese. Like there's be a lactic acid bacteria. In short, that is called as LAB. This bacteria will be used. Basically, it will be added to raw milk, and thereby it will be used to make cheese. Also, making of cheese takes a significant amount of time. It takes a significant amount of time. Getting it? So basically, what
Yes,
will
sir.
happen there? What will happen? You would takes initially you would take some milk. Initially, you would take some milk. You would add lactic acid bacteria into it. Thereby, it will turn into curd. Right. Furthermore,
Yes, sir.
you'd be adding salt into it. Huh? You'd be adding salt into it, and you'd be adding a multiple other things into it, and gradually it will convert into salt. So namely the microbe that would be used for the production of cheese would be the lactic acid bacteria. Keep that in mind. By the way, this lactic acid bacteria which we are referring to, can you guess the name of the bacteria?
Like for lactic acid.
Huh. See, basically, it has got a long name, but basically it's lactic acid bacteria. Just remember this thing. Okay, no?
Okay, sir.
So here, basically, what will happen? We know that milk contains lactose in it. So this bacteria, that is, you have added lactic acid bacteria into it. So it will convert lactose into a lactic acid.
Sir, can I just know the name of that bacteria?
This one, lactic acid
Yes,
bacteria.
sir.
Okay, what is the name of this bacteria you are asking?
Yes, sir.
Okay. See, this simply, the whole name is simply Alabi, I think. That is simply called as lactic acid bacteria. I don't
Oh,
think it
okay,
has
sir.
got any other name. Later
Okay,
on,
sir.
that is the step number 1. In step number 2, what we do, we would be using another bacteria. Although this is not important from the examination point of view, I'm just trying to explain this is how it is done. Okay, now.
Okay,
So
sir.
from examination point of view, you don't have to memorize it. There's a bacteria called as it's propionibacterium shermani. That is propionibacterium shermani. It would be added. Now this would be added to this further. Right now. So this it is added after having added lactic acid body, and now this will be added. What this does now? Now this bacteria converts lactic acid to propionic acid. Propionic acid and release carbon dioxide gas. Release carbon dioxide gas. So in the cheese, the holes that you see in the cheese now, like if you were to see the cheese, there would be so many holes in the cheese now. Like it kind of looks like this. See that this is a slice of cheese.
Yes, sir.
There would be so many holes in it. Why there are holes in it? Because of this carbon dioxide gas. Are you
Okay,
getting
sir.
it? So
Yes, sir.
because of formation of this gas, there are so many holes into it. So carbon dioxide gas is trying to escape from the cheese, and while it's
Oh,
trying to escape from the surface of cheese, it forms the large hole. So you can write it down somewhere. Carbon dioxide gas, while escaping from cheese, makes holes in it. Hope that is clear to you. So that is the whole procedure of how cheese is manufactured. Although like there are
okay, sir.
different types of cheese also, like you have um, different varieties of cheese are also there.
Yes,
So
sir.
different types of bacteria can be used. Usually this type of bacteria is basically used.
Okay, sir.
Now let us learn about some other uses also. Like uh, the bacterias, they would be used in manufacture of antibiotics also. Like in the pharmaceutical industry, antibiotics is also one such medicine. Have you heard of antibiotics before? There are so many common antibiotics we have at our home as well.
Yes, sir.
So it's a type of antimicrobial medicine, and that would be acting only upon the bacterias. Right now, like antibiotics are not given when the person is suffering from a viral infection.
Yes, sir.
So, for example, like common cold or influenza, no antibiotics are given in that case. So you have so many antibiotics. Usually, the common antibiotics you'd have heard, like penicillin is a antibiotic. Huh?
Yes, sir.
Penicillin is one of the most common example of the antibiotics. So they would be antibiotics would be given to treat you or to prevent you from some type of bacterial infections, and they are not going to be effective against the viral infection, such as like common cold uh, or influenza. So hope you are getting it. Have you
Yes, sir.
noted this down?
So 1 minute, sir, please.
Okay.
Yes, sir, done.
Done? Good. Let us learn about antibiotics. Or see, we have only 2 minutes remaining. So how about we discuss few questions from what we have discussed so far?
Okay,
Okay,
sir.
no? Okay. See question number 2. Answer
Okay,
this
sir.
one. Hmm. One of them, we took one of them as an example of a bacterial disease.
So the second one,
That is tuberculosis. Okay, no?
tuberculosis.
Tuberculosis, very good. Answer question number 3.
Rhizobacter bacteria.
That is rhizobacteria. Good.